Welcome back to AllAboutRx. Today, we are breaking down one of the most fundamental metabolic pathways, glycolysis, also known as the Embden-Meyerhof pathway. If you've ever wondered how your cells extract that initial burst of energy from your food, well, you're in the right place. We're going to make this complex biochemistry clear, approachable, and easy to retain for your exams. Here is our macro to micro roadmap. We'll cover the glycolysis overview, aerobic versus anaerobic pathways, the 10-step pathway, the 3 gatekeeper enzymes, the net energy yield, and finally, the clinical significance. Section 1. The glycolysis overview, mapping the pathway. At its core, glycolysis is the main route for glucose utilization in your body. We start with a single glucose molecule, and through a series of 10 reactions, it's broken down into 2 molecules of pyruvate. The key feature you absolutely need to remember here is that this takes place entirely in the cytosol of the cell. And the ultimate goal of this ancient energy-yielding pathway is simple: to generate ATP. Section 2. Aerobic versus anaerobic pathways, oxygen's impact. Now, what's really interesting is how oxygen availability totally determines the fate of that pyruvate we just created. Let's look at the anaerobic pathway first. If there's no oxygen, pyruvate is converted to lactic acid right there in the cytosol. This is actually crucial because it reoxidizes NADH so the pathway can keep running. But if oxygen is present in the aerobic pathway, pyruvate converts to acetyl-CoA, which then enters the citric acid cycle, or TCA cycle, deep inside the mitochondria. Section 3. The 10-step pathway, the chemical cascade. Okay, let's trace this beautifully detailed chemical cascade. Glucose enters and is immediately phosphorylated into glucose 6-phosphate, which traps it inside the cell. As you follow the 10 steps, you'll see that 6-carbon molecule eventually split into 2 3-carbon units. We see our first energy-conserving step, substrate-level phosphorylation, where ATP is actually produced, all the way down to our final product, pyruvate. It's an incredibly elegant, highly organized sequence. Now make sure to screenshot this summary table for your study sessions. While there are 10 steps, you really only need to memorize the 3 gatekeepers, the irreversible, rate-limiting steps. First, hexokinase, converting glucose to glucose 6-P. Second, phosphofructokinase 1, or PFK1, which is the major rate-limiting enzyme. And third, pyruvate kinase, the final step committing the molecule to pyruvate. Section 4. The 3 gatekeeper enzymes, control and regulation. So your body strategically places stoplights along this pathway. Hexokinase, PFK1, and pyruvate kinase act as metabolic valves because their reactions are irreversible. They're controlled by molecules binding to them to either speed them up or slow them down. For instance, hexokinase is actually inhibited by its own product, glucose 6-phosphate. But PFK1 is truly the star of regulation. It's activated by AMP and fructose 2,6-bisphosphate. Think of it like this. When your cell is low on energy, AMP rises, basically shouting, "We need energy, speed up glycolysis." On the flip side, it's inhibited by ATP and citrate. If you have plenty of ATP, these molecules tell the pathway to just slow down because we have enough energy for now. And we can't forget about the liver. It's the master regulator of your blood glucose, using unique enzymes to do the job. It uses an isoenzyme called glucokinase, which is actually induced by insulin. Plus, it contains glucose 6-phosphatase. This is huge because it allows the liver to hydrolyze the phosphate off and release free glucose into your bloodstream to feed the rest of your body. Section 5. The net energy yield, the ATP balance sheet. The entire purpose of this ancient pathway really boils down to one thing: the energy payoff. Looking at the balance sheet per mole of glucose, we actually consume 2 ATP up front. It takes energy to make energy. But we directly produce 4 ATP later on. And the real game changer is the NADH. If oxygen is around, that NADH goes to the electron transport chain. So under aerobic conditions, when oxygen allows that NADH to be fully processed, that gives us a massive net yield of 8 ATP per glucose molecule. Contrast that with anaerobic glycolysis, which produces a net of only 2 ATP per glucose. It's a way lower yield for sure, but it is incredibly fast and doesn't need oxygen. Section 6. Clinical and metabolic significance, why this matters. So, why is this so important for pharmacy and medicine? Well, tissues like red blood cells, the retina, testis, skin, and the renal medulla rely on this as their primary energy source. Why? Because they either completely lack mitochondria, like mature RBCs, or they just don't get enough oxygen. This emergency energy pathway is literally their lifeline. Without those two net ATPs, these tissues simply couldn't survive. Okay, let's do a quick recap of the absolute highest yield takeaways. 1. Glucose converts to 2-pyruvate via 10 steps in the cytosol. 2. Your three gatekeepers are hexokinase, PFK1, and pyruvate kinase. 3. PFK1 is activated by AMP and F26BP and inhibited by ATP and citrate. And 4. Aerobic gives you 8 net ATP while anaerobic gives you 2. I want to leave you with this provocative thought. What would happen to the retina, or your red blood cells, if anaerobic glycolysis suddenly halted entirely? It's a massive cellular energy crisis waiting to happen. That's a high-yield summary of glycolysis. Make sure you screenshot that summary table, and don't forget to subscribe to AllAboutRx for more pharmacy and medical school breakdowns. See you in the next explainer.